Thank you so much. Hi, everyone. I'm Pratik Singh. I'm from Oxford and Technology. And in this talk, Caroline from Imperial and I will give a joint presentation on the talk titled A Multi-Omics Approach, Nanopore-based biomarker detection using aptamers. So in the first section, I'll be giving a brief introduction to Oxford Nanopore technologies. And I'll share a few slides highlighting two potential aspects. One, how the Nanopore platform might be potentially interesting for aptamers and the aptamer community. I say potentially because I don't really have a solution right now, but I don't see why not. And the second part I do want to emphasize is the second part of the talk will be on why Oxford Nanopore technology is interested in aptamers, and I don't see why not. Caroline's talk will be showcasing or emphasizing that exact part. So to start with, I just want to start with the vision of Oxford Nanopore technologies. So the vision is to enable the analysis of anything by anyone, anywhere. So that's our vision. Some of it has already been showcased by our community where they have started sequencing in space, you can see here in the International Space Station. And part of what we've accomplished so far could not have been done without collaboration. And part of what we want to continue in the future will not be established without collaboration as well. So as you can see here, we have been established for 20 years now, and in the past 20 years, we have funded and collaborated with lots of universities and research institutes all over the world. And with that collaboration, we have licensing agreements with more than 40 institutes, and with all the global collaboration and research that we have accomplished with our collaborators, we've gathered more than 2,500 patents and patents applications. So all of this has led to us building a commercialized platform that can sequence DNA and RNA as it stands. And that has been showcased by the community where we have published more than 13,000 publications just using the technology. So how does the technology work? So the heart of the chemistry of the technology is basically a protein nanopore that's embedded into the membrane. So what happens is we apply electric potential, and the ions go through the nanopore to generate an ionic current. So then we add our analyte of interest. So the one that's shown here is the DNA. The DNA is pre-ligated to a nanopore adapter. The nanopore adapter contains a protein, as you can see here. It's a motor protein that unwinds the DNA as well as controls the movement of the DNA going through the pore. So in simple terms, what happens is you have an ionic current that's flowing through the nanopore. The DNA goes inside the pore and blocks the ions passing through. And then there is a drop in ionic current. The bases that are present inside the nanopore, so the DNA bases that are inside the pore, determine what is the current or the block level of this nanopore. And each move of a DNA base going through the nanopore will change the current signal. So this is then deconvoluted to from the raw signal to the nanopore signal using machine learning algorithm, or what we call base calls. So using this chemistry, we have iterated quite a lot. And as it stands, we now have a technology that can base call using our platform to an accuracy of 99.75%, which is P26. And we have a robust platform that can do high throughput sequencing and give you high output. Why is it important in this case is, so the technology is read diagnostics, which means we can sequence DNA from 20 bases, as less as 20 bases, to as much as megabases long. And we don't see why this technology cannot be used right now to sequence DNA aptamers, single-stranded DNA aptamers that are as short as 20, 30 bases, or it could be longer to 100 bases. What makes it more valuable in this is because we are reading the DNA in its native form, we can simultaneously detect modifications, not only the 4 canonical bases, but all the modifications that are in the DNA in real time without the need to do any additional or analysis. So far, what we have trained using our machine learning model is just the genomically interesting modified bases, such as 5MC, 5MC, 4MC, and 6MA. That's when I come to a potential thing on potential application where I see a lot of exciting research where you modified the DNA aptamers to improve binding. And right now, the way to detect those binding modified aptamers is using Illumina or deep sequencing using NGS and then converting the data based on which base could be modified or using mass spec. It would be possible to use our platform right now by giving information on those modified bases that are very interesting to generate novel aptamers that have binding efficiency and train a model to base call it in real time so that once you have an enriched pool of aptamers, you can use that pool to sequence in Nanopore and get the exact sequence of the DNA bases. Same thing can be applied for direct RNA as well. So right now, our technology is the only one that can do direct sequencing of RNA bases. So you don't need to reverse transcribe to make it a cDNA and sequence it. And same thing with direct RNA, we also have been able to detect modified bases in our case. And right now, what we have done, as I said, is again with genomically interesting bases. I don't see why we cannot include different RNA mods that are being used in aptamers to detect using our platform. So that's where we stand right now. What's next for Nanopore in terms of future applications? So Nanopore is heading toward multi-omics. So not only DNA and RNA. What we are looking for is to be able to combine and analyze different data sets to get a more comprehensive understanding of biology. And why is Nanopore interested in multi-omics? Is basically we want a single platform that can determine or that can detect or analyze all these different data types more than just DNA and RNA. And the next two things that Nanopore is interested is proteomics and metabolomics. So in proteomics now, we are doing single molecule protein sequencing using the Nanopore platform. So this is an active research that has been in Nanopore for a few years now. And we will be showcasing some of the advances that we have done in protein sequencing in London Calling, which is next month. So London Calling is a conference that's usually hosted by Doctoral Nanopore Technology every year. So it's an annual conference where we give updates on what's going on, like new and exciting things that's going on in research. So this is a conference that you can attend online, and it's free to attend offline as well. So any updates will be here. And the next thing that I want to say is the new project that we have started in Nanopore is metabolomics, which is single molecule metabolite sensing. And that's where we are very interested in aptamers because they are amazing sensors or amazing binders. So we want to use as aptamers as a potential intermediate sensor to detect a small molecule or a diverse set of small molecules and use the Nanopore platform to be able to detect it. And that's where Caroline is going to showcase some amazing data on how she's used aptamers and the Nanopore platform to detect different biomarkers. Okay, sorry.
Okay, thanks, Pratik. Well, I'm Caroline. I'm currently based at Imperial College and collaborated with ONT for the past around 6 years, first during my PhD, now during the fellowship. And I'm going to talk to you about how I use the ONT technology, which is generally long-read sequencing, to a more sensing-based approach. So we want to develop an assay for biomarker detection towards clinical applications. So we've heard a lot about biomarkers already. Biomarkers indicate diseases. So we want to detect them at low concentrations, basically when people don't show symptoms yet. And we also want to use liquid biopsies like urine and saliva that are non-invasive. So we want to use a sensor that operates in that area. Low concentration and non-invasive. So we thought nanopores are a good starting point for that because it's single molecule sensors with great sensitivity. So the aim really is to build an assay that can detect low concentrations of biomarkers and ideally in a multiplex way. So we want to look at multiple biomarkers simultaneously in order to increase the specificity for a disease. Because yesterday we talked about that as well, that just looking at one biomarker is not specific enough really to indicate the disease. So we really want to fingerprint what's going on in a patient's body. So the alternative approach that we've come up with is looking at biomarkers such as microRNAs, proteins, and small molecules in serum. we use the ONT technology, so the benign flow cell with barcoded molecular probes, which I'll introduce in the next slide. And then we look at the electrical current readout that indicate the presence and the concentration of those biomarkers in that sample. So this whole assay from sample preparation to result takes about 60 minutes. The probes mainly consists of 3 regions. The first region is the adapter. Pratik mentioned that in one of his slides. That's the enzyme that controls the speed of the translocation. We need that in order to read the bases. The DNA barcode sequencing region, which is a 35 base sequence. It's random, but that is the bit that is actually sequenced. That basically indicates the identity of the probe. And the target binding region, which is either a complementary microRNA region sequence if you want to detect microRNAs, or an aptamer if you want to target proteins or small molecules. So the way this works, you have the nanopore with the motor protein sitting on top. The barcoded probe translocates through, and you read the barcode, so that blue bit. The barcode gives you a unique current signature that again gets base called into the sequence, and that's what we use to identify the probe. So we've made many of these probes, 40 in total for this publication. So you would align the sequenced event against the references library, and basically the one that gives you the highest alignment score indicates that this is that barcode. So here I would show barcode 37 with the highest alignment score, so we can be sure that this actually is barcode 37. So we did that with all the other probes as well, showing that we can identify all of them, align them to make sure that when we say this is barcode 1, it's actually barcode 1. So this is the first step. Now, shifting gears, we add targets to this. So we have either a microRNA, a protein, or a small molecule. The microRNAs, they form a double strand, and because it's too large to fit through the pore, it has to unzip. Same with the protein or the small molecule. So we see a delay in that electrical current. So we basically get a binary response. So is the event delayed? Yes. So the biomarker is present. If it's not delayed, the biomarker is not present. So the barcode identifies the probe, and the delay indicates whether that biomarker is present or not. So we've done this with 40 barcodes and 40 microRNAs in a multiplex experiment. I'm showing here the average signal. So on the x-axis is the concentration, ranging from 50 picomolar to 50 nanomolar. And here is the normalized percentage delay. So the events that were delayed divided by the total number of events. So now with that technology, you can demultiplex the signal based on the barcodes that we have, generating 40 individual titration curves just from a single experiment. And these titration curves then can be used to indicate the presence and the concentration of biomarkers of an unknown concentration in clinical samples. And we validated that with RT-qPCR. So that was synthetic targets. We've done this in serum as well. You get the idea. So we looked at 40 participants. Those were healthy participants. So this is not clinically relevant at all. We just wanted to show a proof of concept that we can detect microRNAs directly in serum. Showing upregulation for some of those microRNAs that again we validated with RT-qPCR. Basically the same technology we use for protein and small molecule detection. We chose 4 different proteins, serotonin is the small molecule. We tested a bunch of different aptamers. Most of them didn't work, and some of them did. So we looked at the translocation time compared to the control. So we just have the probe without the target present. So the translocation time of the probe going through is much lower compared to when the protein is present, because that unraveling of the probe takes time. And then we do the same thing with the percentage delay. So we look at how many events are delayed, showing a significant increase in percentage delay for when the proteins are present. And the concentrations we chose were clinically relevant. So these are all, proteins related to cardiac fibrosis, and the concentrations were relevant for that disease. And then taking it one step further, we were interested in combining this, so combining microRNA protein and small molecule detection. Again, this is not clinically relevant, but a proof of concept that we could actually do this. So we chose a microRNA from the serotonin, and then added this at different concentrations in a multiplexed experiment. So we compared it to the control when these targets were not present, added a low concentration of these biomarkers showing a percentage delay increase, even higher concentration and even higher increase for that. So that was very promising, because for now we need to use different technologies to detect these different biomarkers. And then lastly, this is kind of what I'm working on at the moment. It's a more multi-omics based approach. Like I mentioned, ONT is more known for long-read sequencing. What I've shown you was sensing, but now the question is, can we all bring it together to detect, but also sequence at the same time? So we would use proteins, something like sulfur DNA, microRNAs, at these probes that I've shown you. They are basically then marked with the barcodes, and then we would use the ONT readout to do sequencing and sensing at the same time. So I've chosen a DNA sequence on that DNA and one of the microRNAs. I added this, and I know this looks very messy, but if you look at this long enough, you see a pattern, and if you don't, we use machine learning to do that for us. So we have the gray bits here, which are just the barcoded probes, so they didn't bind to the microRNA. The microRNA in green, those are the events that show a delay, and then the long events are the DNA events. So we have 3 different populations essentially, but these two, they basically have the same length because it's the barcoded probe. So we get this histogram, and we clearly see 2 populations, which is the sensing population that has the barcoded probe that detects the microRNA. And then we have the sequencing population, which is around 3.6 kbp, which is the lambda DNA that I used as a proof of concept. And then you can look at the sensing population and pull out that delay data to see whether microRNA is present or not. And so that shows successful sequencing and sensing in just one experiment. So in conclusion, I've shown you that we've detected 40 microRNAs simultaneously with synthetic targets, but also in serum. We sensed serotonin and proteins, and with the goal to move us closer to a more multi-omics-based approach, personalized medicine. So for the future, there's lots I want to do, but for this audience, what I really want to do is optimize the aptamers to detect the proteins and the small molecules for this type of assay. So I want to thank my mentors, Dr. Edmund and Alex at Imperial College, my students and collaborators at Imperial, and of course, ONT for supporting me throughout all these years, and you for your attention.